Welcome once again. I'd like to welcome Professor Christoph to this talk. He's the presenter for today, and he's going to talk about stable isotope labeling of mammals. Um, Dr. Turck has a very prestigious, research pedigree. He has done his, bachelor's in University of Aachen, Germany, followed by PhD in peptide chemistry. He spent a lot of time in the US working for Roche and also working at UCSF as the director of protein structure laboratories. And later on, he went on to work as faculty for, um, you know, various different universities, Ludwig Maximilian University at Munich, and he currently serves as a professor and director at Chinese Academy of Sciences in Kunming Institute of Zoology as well. Thank you so much, Dr. Chris, and please go ahead.
Yeah, thanks a lot, Ravi, for the nice introduction. I apologize for the technical issues I have here.
That's okay.
Maybe the people in academia will appreciate that, but Teams is too complicated.
It's hard, I know.
I'm a Zoom person. Anyway,
Yeah.
I'm going to give it my best. I hope you can all see
Thank you.
the slides. So thank you for giving me the opportunity to showcase our SILAM projects over the past 20 years, actually. And it's interesting. I'm on the next slide. This is actually the first publication on stable isotope labeling of mammals, or SILAM for short, which was developed in John Yates' lab at the Scripps Research Institute. And it's interesting that it has its 20th anniversary this year. Now, originally, this was developed for mass spectrometry purposes for relative quantification. The idea was, just like with cells or smaller organisms, to see if higher organisms, specifically mammalian organisms, were able to use to incorporate 15N and the resulting specimens, tissue, plasma, whatever, could be used as a reference material for relative quantification of proteins by mass spectrometry. And it turned out this worked really well. Also, one question, of course, at the time was, are there any ill health effects that 15N could cause? But apparently that was not the case. And this was actually supported by our data that we also developed our food, our own diet with Silantes. We didn't see any ill health effects. The extra neutron in these in the nitrogen nucleus apparently does not is of any harm. And I'm going to get back to this actually at the end of the talk when I talk about the isotope effect. So this is sort of the outline of my presentation today. I will tell you initially something about relative quantification of proteins by mass spectrometry, just like the Yates lab was doing in the beginning. Then I'm going to switch a little bit to not looking at the static proteome, in other words, not taking snapshots, but we got interested in protein turnover, which appreciates a lot more the dynamic nature of the proteome. As we all know, unlike the genome, which is static, the proteome is a dynamic entity. And actually, in order to understand it, it's not enough to take a photo, a snapshot photo, but you actually have to take a movie because it's such a dynamic entity. In that, in the same vein, we also looked at long-lived proteins. It's a study we just published this year. And then I'm going to switch briefly to other applications that you can utilize SILAM specimens for, and that's metabolomics and stable isotope effect. We briefly touched upon. And at the very end, I'm going to tell you about a new product that Silantes is about to release, where we're not labeling mammalian organisms, but mammalian cells in culture. So just by way of introduction, this is where this Yates paper came out. It was actually a great advantage for us because we were working a lot on mouse models for psychiatric disorders. It was about the time when I joined the Max Planck Institute of Psychiatry in Munich. And we teamed up with Silantes and Hermann Heumann, the CEO of Silantes. We asked them, well, can you also develop, in this case, a mouse feed that allows us to 15N label mouse models? And Silantes came up with a great idea and feed that is based on Ralstonia eutropha, which is a small organism, bacteria that you can efficiently label. And it served as a protein source for this mouse diet. So you culture these bacteria, make them 15N labeled, almost 100%, and then you hydrolyze the proteins. And this is basically what serves as the amino acid and protein diet in the Silantes food. So since the major nitrogen source in this feed is nitrogen, you can actually get very high incorporation rates this way. And this is something I'm going to show you here on the next slide, how we went about labeling these mice. We already fed the mother mouse upon pregnancy detection with the 15N labeled food from Silantes. So the embryos, of course, are already being exposed to some 15N. And once they're born, they still rely on the mother's milk, which at that point also has 15N labeled bacteria, 15N labeled proteins. And then upon weaning, about here postnatal day 14, the pups, the offspring starts feeding on this diet, and we fed them for about 2 months, and at postnatal day 56, we harvested all the organs and the blood, and you can actually take advantage of everything that this mouse has because everything is now at 15N labeled. And just on a side note, all these organs, 15N labeled, are available at Silantes at the current time. Not only the feed, but also labeled mouse tissue. Now, what you see here on the right, the bar graph, is the 15N incorporation rates that you get over time, starting at postnatal day 5, 14, 28, and 56. You can see that the 15N incorporation gradually goes up. And in this case, we actually compared the Silantes diet to another diet that was based on algae, also 15N labeled algae, and we got pretty much comparable results with the algae diet at the time. In order to analyze the different incorporation, we developed, we is Katrin Heckler, a student at the bioinformatics student in the lab at the time. She developed the so-called QuantiSpec software, which uses MALDI time-of-flight data, mass spectrometry data, and compares the acquired isotopal patterns from tryptic peptides to theoretical peptide isotopal patterns reflecting different 15N incorporation rates. And this was very important for us, of course, to have this software available in order for us to ascertain that at postnatal day 56, we're dealing with material which is over 90% labeled. So a very efficient way of doing this with the Silantes diet worked out very well. One thing, though, that we realized, and everybody else realized too, of course, you never get 100% label. 100% label, maybe if you do many, many generations, you can approach 100%, but you're probably never gonna get rid of some of unlabeled or 14N isotope. Why is that? Well, in the diet, of course, there is not only protein, which might be almost 100% labeled. You also have other compounds like vitamins and other small molecules that have nitrogen, which were not labeled. So as a consequence, and that's shown here on the right, if you look at the mass spectra, if you know something about tandem mass spectrometry with data-dependent acquisition, what happened at that, well, still happens today, the most intense isotopologue pattern is taken for fragmentation. And since you're dealing here with peptides that are not 100% labeled, the 15N monoisotopic signal, which is the peptide that has only 15N, is not the most intense signal. What is the most intense signal for most of the peptides that we were dealing with is the -1 Dalton isotopologue, where basically one nitrogen is not 15N, but 14N, okay? So this was the isotopologue that was mostly taken by the mass spectrometer for fragmentation. And as a consequence, the number of identified peptides was not really optimal. And I had a very talented graduate student in my lab at the time, Yaoyang Zhang, and I'm actually still collaborating with him. He has his own lab now at the Chinese Academy of Science in Shanghai. He came up with a nice idea of how to improve this situation by basically telling the database search engine to look at the -1 Dalton signal for protein identification efforts or peptide sequence identification efforts. So that, if you introduce a -1 Dalton static modification during the database search, then you actually can vastly improve the number of identified peptides this way. So I'm telling you all this that, because, when you do SILAM, as you go along, like in science always is the case, you sometimes have to develop certain tools to optimize the method. But this worked out really well for protein identification. Now, so this is the also an important aspect to consider. What you should not do if you have 15N labeled material at your disposal, and you want to compare two conditions, control and case, let's say an untreated animal and one treated with a certain drug, you should not directly compare them. In other words, you should not mix plasma, for instance, from the unlabeled control animal and plasma from the 15N labeled case animal, because you can never exclude, first of all, an isotope effect, and secondly, that the diet, the 15N diet, might be a little different from the unlabeled diet. So what you do, and everybody does this, of course, you use the labeled material, the 15N labeled material, as a reference. You do 2 rounds of mass spectrometry. In one case, you mix the control specimen with the 15N specimen, and in the other case, you mix the case specimen with the 15N specimen. You combine these, be it plasma, be it tissue extracts or whatnot. You can do some protein fractionation. You digest your proteins, and then you do tandem mass spectrometry. And so the beauty of this, of course, is that once you have these mixed, you don't have to worry about any artifacts, a loss of one sample, the controls, the unlabeled sample and the labeled sample, because everything is in one pot. But at the end, what you do is you get 2 mass spectra here for each peptide, one of unlabeled control and reference, and one for the case and reference. And what you have to do for relative quantification, you have to form the ratio of ratios. So you form the ratio here for the control and the ratio for the case. And then the ratio of ratios gives you the relative amount of this particular peptide and protein between control and case situation. So this worked out really well. We've used this extensively for many mouse models. Just on the side note here, why we were interested in mouse models. Well, mouse models, unlike people, are inbred. You can get them as inbred strains, which means they have a very pretty much identical genetically they're identical, which leads to a very homogeneous phenotype. You can also house them over a very controlled environment with regard to food and sleep and whatnot. You have a pretty decent number of animals, and you can do metabolic labeling All of this, of course, not the case if you start with patient specimens. So we did a lot of work with mouse models. This is sort of like the strategy we were pursuing. You start with a very complex disease phenotype like depression. We teamed up with a very talented group of behavioral scientists at our institute that modeled certain aspects of these very complex disorders. You can never model this, depression in a mouse, but you can model certain aspects of these very complex disorders. And then we came in, did our biomarker studies by mass spectrometry with the objective to elucidate pathways and networks, molecular networks that are affected in these phenotypes to get a better understanding of the pathophysiology. And the ultimate goal, of course, was to translate these data to the clinic. And I'm going to just present one example here of how we went about this. So in the next slide, of course, you can appreciate that you get big data. You get a lot of upregulated and a lot of downregulated proteins. And you need to resort to bioinformatics to make sense out of that data in order to delineate pathways that are affected in this particular phenotype. Now, one example that I'm going to show to you here is a project that we engaged upon that has to do with the antidepressant response. This is actually a major issue for psychiatry in the clinic, that many people do not respond to the currently available antidepressants, which are mainly selective serotonin reuptake inhibitors. And we wanted to shed some light on pathways that might be involved in response or non-response towards these drugs. And for this purpose, we used mice again, and we fed them chronically paroxetine, which is a selective serotonin reuptake inhibitor. We did that for about a month. And then we found indeed, by using a lot of, over 100 mice, we found indeed that we can distinguish so-called responder mice and non-responder mice. Now, how did we distinguish those? This is a behavioral assay that's known as the forced swim test, where the mouse has to swim or in a beaker of water. And the quote-unquote depressed mice have this tendency to just float. In other words, they're very passive, which is a kind of a hallmark of also depressed patients. And just to make a long story short, we found the majority of these mice were responding, were struggling when they were exposed to this forced swim test, but there was also a small subgroup that was not responding. And in order to make sure that this has nothing to do with the amount of drug in the brain, we actually took some mice and measured the drug in the paroxetine in the brain and found that the levels are pretty much the same. So this response or non-response has nothing to do with the amount of drug that these animals had in their brain. Then my graduate student at the time, okay, I see some questions popping up. Maybe we'll wait until the end, Ravi, for answering these questions because of these technical difficulties.
Yep, that sounds good. No worries. I just asked the attendees to just type in anyway, but at the end, I'll ask those questions to you. No worries. Thank you so much for asking questions, guys. Appreciate it.
Yeah. I'm a little nervous about, with the technical difficulties to end the presentation.
Yeah, I understand. Yeah, please go ahead.
Yeah, so no worries. We'll get to the questions again. So anyway, in this case, the mice were then, we took the hippocampi of these mice. Hippocampus is involved in depression. That's been known in patients and also in mice. Now, and then my student at the time, Dong-Ek Park, he did extensive proteomics and metabolomics. And just to make a long story short, he found again a lot of up or down regulated protein expression. Whatever you see highlighted here with colors, these are results from proteomics and metabolomics. These are proteins or metabolites that are part of interesting pathways that I'm showing on the next slide. It turned out that two pathways, namely purine metabolism and pyrimidine metabolism, seemed to be the most affected in the hippocampus of these mice and were different between the responder and non-responder mice. So this is an interesting finding. We not only found this in the hippocampus, but we also found this reflected in the blood, in the plasma, which gives us great hope that this could be eventually be used for a clinical assay. Because if you're dealing with patients in psychiatry, you of course cannot take any brain biopsies that cannot be ethically justified. What you're relying on is of course peripheral fluids, cerebrospinal fluid, or ideally blood, and develop a blood test. So the good news here was that the differences that we found were not only present in the hippocampus, but also in the plasma. And Dong actually took it a step further, and it was the great thing about being at the Max Planck Institute of Psychiatry. We have an associated clinic, where patients are being admitted, and they're also being subjected to blood assays. And there's this Munich MARS, or Munich antidepressant response signature study. And we got samples, blood samples, PBMCs, so peripheral blood monocytes, mononucleotides. And Dong then looked at the expression of these proteins that he had found in the mouse to have altered expression levels, and found a very good correlation between the expression of one protein here from the purine metabolism and one from the pyrimidine metabolism, that where the expression nicely correlates with the clinical response of the patient, who was also subjected to SSRI treatment. So this is sort of like a tiny little step towards clinical translation, which of course was always on our agenda. Working with mice is fine, but eventually, of course, you want to translate this to the clinic for the benefit of the patient. So this worked out really well. Okay, I'm going to shift gears now and shift to, as I mentioned in the beginning, to some projects that have to do with protein turnover. Again, the proteome is a dynamic entity. What is protein turnover? Well, it's the combination of protein synthesis and degradation. There's several scenarios. Either you have an equilibrium where the protein amount doesn't change. You can have a pool expansion by either increased synthesis and unchanged degradation, or vice versa, unchanged synthesis and decreased degradation. And then the opposite is true for pool contraction. This is basically what's behind protein turnover. Now, we had to change our labeling strategy, of course. We could not do this with fully labeled mice. But what we did instead is we took adult mice, feeding them first with unlabeled diet, also to adjust them to the diet, the Ralstonia eutropha-based diet, where you have the pre-existing unlabeled protein. And then we call this day 0 here. We start feeding them with 15N Silantes diet. And from then on, nitrogen 15 and nitrogen gets incorporated into proteins and also other molecules, of course. Now, we then took this tissue again and did two types of mass spectrometry. I'm going to explain tandem mass spectrometry. The other one is called MIMS. I'm going to explain that in a second, what this is. Both of these methods gave us very valuable protein turnover information. Now, this is sort of like what you see as you go along. Again, we had to develop a software. I had a number of students, bioinformatics students, and also Yaoyang Zhang was again involved, who developed the so-called ProTurnizer software. Now, the ProTurnizer software interrogates these very complex isotopologue patterns from tryptic peptides. And what I'm trying to illustrate here is these kind of yellowish signals that you see. This is unlabeled peptide. And the purple signal is the labeled portion of the peptide. So it's identical peptide, just with different numbers of 15N. In the beginning, you have almost no 15N, and as you go along from day 1 all the way to day 7, this happens to be a protein with very high turnover. The purple 15N isotopologue signals increase. Now, there's vast differences, as you can imagine. I'm showing 2 examples here of tryptic peptides. One of them is from DPYSL, important protein brain development. The other one is heat shock protein 71. So again, this is coming from the hippocampus of mice that were labeled for about 2 weeks with the 15N labeled diet. And as you can tell, there is very big differences in how much 15N is being incorporated in these 2 proteins. So very big differences between protein turnover. The software, by the way, relies on the assumption, and that's something we proved, that the 14N monoisotopic peptide signal mainly originates from the pre-existing protein population. That's an important assumption in this case. Now, Yao Yang did this also extensively again for brain and hippocampal proteins. And he says kind of a summary of the results that he got with regard to what molecular pathways are very active in the brain and which ones are less active with regard to protein turnover that happens in the brain. So very valuable data about brain physiology. Now, one interesting example we stumbled upon, and unfortunately we couldn't actually resolve it totally at the time. This is a protein family known as DPYSL, which is important for brain development. So there's 3 different family members that I'm showing you here. And as you can tell, this one down here experiences a lot of turnover. We have a pretty significant 15N signal, whereas the other 2 have a rather small 15N signal. That was already one interesting result, but what was even more surprising, maybe not, but interesting for me, and still wish we could have followed up on this, in the 2 DPYSL proteins that have a low turnover, we see 2 populations. Okay, here you can see it, and also here. This is not like a nice little envelope. There's sort of like 2 envelopes that you can detect. So the hypothesis is, or the assumption that we have is that we're dealing with different turnovers of the same protein, either in different brain regions or maybe in different brain cells. The brain is, as you all know, is the most complex organ. There's thousands of different cell types in there. And you could easily imagine that, for instance, in neuronal cell and a glial cell or astrocyte may have different turnover mechanisms. The way you would approach this today is, of course, you would do single cell proteomics. You would isolate, neuronal cells, astrocytes, glial cells, or whatnot, and then see if you can actually untangle these 2 little bumps here and which turnover belongs to what cell type. But very interesting result that again emphasizes the fact that the brain is so complex, right? It's not like a liver where you probably have, have only pretty much one type of cell and probably one type of turnover. In the brain, everything gets a lot more complex. Okay, at the time also, I was fortunate to team up with another researcher. His name is Claude Lechen at Harvard University, who's also interested in stable isotope labeling of tissues and protein turnover. And he uses a different type of mass spectrometry called multi-isotope imaging mass spectrometry. He can generate images based on labeled material. The way this works is that you mobilize your tissue slice, a brain tissue slice. It's then bombarded with positively charged cesium ions. As a result, the tissue emits negatively charged cyanide ions. And what this image, with the images that he generates, reflect is the ratio of C15N and C14N signal. So I'm showing you results from a mouse hippocampus. This is a tissue slice that was stained. And in the following slide, you see actually a tissue slice that was generated with the MIMS technology. Beautiful. I still think it's a very beautiful image. You can make out different regions, subregions of the hippocampus here. And the blue signal is part of the hippocampus that has very little label incorporation. This is again from mice that were labeled and fed with 15N diet. And here the CA3 subregion has quite a bit of red signal, indicating that there's something happening in terms of protein turnover, a more active region of the hippocampus. Now, again, we then, since we had both of these technologies at our disposal, we used, again, a study, or got engaged in a study that has to do with the antidepressant response. My postdoc at the time, Michaela Filio, subjected these hippocampi to tandem mass spectrometry, protein extracts, and also to MIMS analysis. And this is probably too much detail here, but she found differences in different brain regions and also in organelles between the treated and non-treated animals, mice animals treated and non-treated with the paroxetine. This is something all published. You can look at the paper to get more information about this. But this shows you the power of these two methods. You're getting molecular data on one hand from the tandem mass spectrometry, and you're getting beautiful image data from the MIMS method. Okay, moving right along, and this also has to do with turnover, of course. And this is a study that we published this year. It has to do with the long-lived proteins. Why is this of interest? Well, protein chemists among you can appreciate this. Proteins, when they get old, they can get damaged. They often get modified by oxidative stress situations. They can be fragmented, and there's many things that can happen to a protein. And that's why protein turnover is so important. If a protein gets modified or fragmented, it loses its function. So the cell, of course, has to make new and fresh protein. And that's why protein turnover is so important. But we wanted to find out, there are some proteins that are very long-lived, which of course could be candidates for biomarkers, because the chances are that they are damaged over time, and depending on the tissue dealing with, they may have potential to serve as biomarkers. Now, in this case, this is some summary of what can happen to proteins. Mainly oxidative stress that is causing these damages. Sunlight, cigarette smoke, and many things from the environment can cause these protein damage. And what we did here, in this case, we labeled these animals even longer, namely 89 days, with 15N labeled diet. And we looked at the end and wanted to identify proteins that still had unlabeled signal. Okay, after 89 days, you get overall, very high incorporation rates, well over 90%. But if you then still find proteins that are unlabeled, this is an indication for a long-lived protein. Now, this is actually a collaboration with Yaoyang Zhang in Shanghai, my former graduate student, now professor in Shanghai. They found a lot of proteins, identified and quantified a lot of proteins, and then found also quite a number of long-lived proteins. These are the different organs that were interrogated. Not surprisingly, the brain, the red squares show or indicate the long-lived proteins. The brain has the most because it's a very low turnover organ. But what was really surprising is this result here, namely the plasma. The plasma had, although we identified the smallest amount of proteins, which is not surprising because the plasma is very hard to comprehensively identify with mass spectrometry due to all the abundant proteins that are in plasma. But surprisingly, we found the most long-lived proteins in there. And that's, of course, a worry, right? If you have a lot of these long-lived proteins in there, they can get, maybe damaged and may, on the other hand, for diagnostics be of value. It turned out that these proteins that were long-lived, we found 3 different pathways where they belong to. Namely, there's a lot of histone proteins. The histones are wrapped around the DNA and they sort of protect it. And they turn out to be very long-lived. And the other ones have to do with proteostasis, namely ubiquitin-related proteins and also proteasome-related proteins. They seem to be the top long-lived proteins that we identified. Many of them turned out in the plasma are related to cardiovascular disease. This is the summary of this analysis, which of course might be good candidates for this particular group of disorders. And more interesting for me, of course, are long-lived proteins in the brain. We found what you see here on the left is the different tissues, and I cannot even read it here on my screen, but wherever you see a red signal here, a red square, and then you look on the right for the pathway, for the brain, you find pathways that actually have something that are correlated, that have something to do with brain disorders, namely neurodegenerative disorders, schizophrenia, and others. And for kidney, we find the right related disorders, and for other organs as well. So this again drives home this message that we wanted to convey with this study, is that these long-lived proteins might be very good biomarker candidates for particular tissue disorders, be it brain or be it the heart or whatnot. I'm going to skip this long-lived DNA and move right along what we now think of doing or have already done to some extent. I mean, the SILAM tissue is so valuable. There's probably many more applications, not just protein identification or quantification and protein turnover. There's probably a lot more things that you can do with it. And one area, of course, that also needs reference standards is metabolomics. Ideally, there you want, if you do untargeted metabolomics, you want to have reference standards for these different metabolites that you're trying to quantitate. And at that time, we had made some attempts to label these mice with 13C. Now, with 13C, even if you have, a protein part of the diet that is over 90%, there's so much other carbon, namely lipids and sugars and whatnot in these diets, that you never get very high incorporation rates. We were able to go up to 60% incorporation with these 13C mice. But we asked the question, can we use this material still as a reference for metabolomics experiment, untargeted metabolomics experiments? So my postdoc at the time, Frederick Detloff, who's now at the Max Planck Institute for Aging Research, he did a dilution series where he mixed human plasma, which is of course of very great interest, with mouse plasma that was partially labeled with 13C. Now, as we probably all know, mouse and human, they're not identical, but they share a lot of metabolites, especially in the blood. So this study certainly had merit. And he specifically looked at amino acids and the 20 natural amino acids, and did a dilution series, and was able to demonstrate that by adding the 13C reference plasma to the human plasma, he could vastly improve the accuracy of the quantification of the different 20 amino acids. So this is something we at Silantes is thinking about to expand upon this. This is a collaboration with Rainer Schumacher's lab at the Boku University in Austria. And we're now following up on this approach because because you have so many labeled reference compounds in the mouse that this could maybe be a very universal standard for many metabolites in these samples. Now, I'd like to finally finally also talk a little bit. This was sort of like a fun project. Touch upon the isotope effect. Now, it's commonly believed that 15N or 13C is not a big deal. There's just one extra neutron. In the nitrogen, you have 7 neutrons. And then in 15N, you have 8 neutrons. Not a big deal, just one more neutron. Doesn't make a big difference. And for the most part, this is sort of like accurate. But if you look closer with a fine comb, you can actually find differences. What we found during chromatography, if you really run very shallow gradients on the reversed phase column, we found that the 15N labeled peptides, tryptic peptides, elute a little earlier than the unlabeled tryptic peptides. And that was true for pretty much all of the peptides of the same sequence. So this was already an indication there is an isotope effect. It doesn't really matter for the relative quantification that you want to do. If you make this window large enough, both of these peaks of peptides are being fragmented. So it's not a problem for relative quantification, but it shows you there is an isotope effect. And we got a little curious about this, and my postdoc at the time, Michaela Filio, what she did, real quick experiment, she used E. coli and cultured them either in unlabeled media or in 15N labeled media, and did find, lo and behold, that the 15N labeled bacteria in the media grow slower than the unlabeled media. Furthermore, she found differences in protein expression between the two E. colis in terms of certain proteins. One particular enzyme that is also affected, and which is validated here also in the Western blot, is relevant for synthesis of pyruvate. So she looked at pyruvate levels between the unlabeled and the labeled E. coli, and did find pretty significant differences. So short summary here, there is an isotope effect. It's not really relevant if you use this material as a reference, but it might be useful for other studies where you can maybe actually take advantage of it. And one very curious experiment that we did, we also compared our unlabeled and 15N labeled mice, found also differences in the serum, protein expression differences, and this was the real surprising result. There is differences in behavior between these two mice. This is again a behavioral assay that assesses depression-like phenotypes. It's called the tail suspension test. We found the 15N labeled mouse to be a lot more active in this assay than the unlabeled mouse. So even behavior seems to be affected by the extra neutron in nitrogen. Okay, I'm going to wrap up now and want to drive home one thing. This again shows you the ingredients of the Silantes SILAM feed. And a lot of people are critical about this method. And one reason is that they say, well, this is very expensive. And yes, the food is not cheap, but you should look at the numbers here. A typical mouse eats about 5 grams of diet per day. Okay. So if you were to label this mouse, feed this mouse for 2 months, this mouse will be worth 5,250 US dollars. That seems like a high number, yes, but I mean, look what you get out of this mouse. You get all these different tissues, and you could even take more. You could take hair, you could take eyes, you could take the tail and whatnot. And these are pretty decent tissue weight numbers. I mean, look here, cerebellum, 60, 80 milligrams. And here the liver, you have 700, almost a gram of liver. So try to think of it, how many mass spectrometry experiments you can do with this. And even if you're only interested in the brain, maybe you have a colleague who's interested in the liver, or someone is interested in heart, you could even share these tissues. And that's why I'm saying, yeah, for this 15N, you get a lot of mass spectrometry for the buck. This is a great, it's expensive, sure, 5,000. But you can do lots of experiments. I would argue it probably lasts you a lifetime once you have a good collection of these tissues, labeled tissues. You probably only have to do this once. Now, finally, then, I'd like to just make you aware of the fact that Silantes has now developed a media. So this is not really SILAM. It's not a mammalian organism, but it's mammalian cells that can also be labeled with 15N. This is the major applications are for NMR. If you do NMR protein structural studies, these proteins have to be labeled with stable isotopes, be it 15N or 13C. And this took about 2 years of development to come up with a very good mammalian cell culture media that also gives very high incorporation rates, over 90% incorporation rates, and can be used for NMR and other applications. And with that, I'm at the end of my talk. I again apologize for the technical difficulties, but I'm sure we still have some time for questions. Thank you.
Awesome. Thank you so much, Dr. Turck. Thank you everyone for attending the webinar. We do have some questions, Dr. Turck. So I'm going to read the questions out loud so that everyone is aware. The first question is: do you observe 14N and 15N peptides, right chain N and amide N, for example? One, precursors and afterward their fragments. Which algorithm is used for quantification?
Well, they both have to be used for quantification, obviously. Comparing basically the 14N signal intensities and the 15N signal isotopic signaling intensities. And that gives you the ratio for control and reference and case and reference. And then again, you have to go one more step. Ratio of ratios gives you the relative quantification between case and control. So yes, you get of course mass spec data for both. You have to do a database search for the unlabeled peptide and also a database search for the 15N labeled peptide of the same sequence.
Okay, thanks. The second one is, please feel free to raise, click raise and this hand raise button so that I can allow you to, ask the question directly to Dr. Turck as well. I'm just going to go through the questions that are in the chat first, so that we're going to go through that, and then I'll allow you to ask questions directly. The second question is: are labeled tissues from different brain regions and are early developmental stages available from Silantes? Can adult labeled whole brain tissue be used as a reference for a different developmental stage or a brain subregion?
Last question, certainly. this reference you can use for anything that has to do with the mouse brain, of course. I guess the question, so the first question is there tissue available from Silantes from early mouse development? The answer is yes, but these early development tissues are not fully labeled. They have small, lower incorporation rates. So that's something you don't want to use for relative quantification purposes. But what you can do, of course, is you take tissues from different developmental stages of the mouse, and then use the adult mouse, so to speak, the developed mouse reference material for the relative quantification purposes. Yes, that's the way I would go about it.
Okay. Yeah, it seems like, every generation kind of labels the different, not different organ, but it goes by, if you want to achieve like complete brain labeling, maybe you need like a second generation or something like that, as far as I know. Okay, third question. Could you detect some proteins half-life turnover during the developmental stages, tissue or organ specific?
The data that I showed you is pretty much only reflecting one stage. So it's sort of, again, a snapshot. You would have to do this for different ages of the mouse in order to get that data. But I would think, if you have turnover data, I would argue that a lot of this turnover is probably the same, doesn't change that much over time, unless there's certain, drug treatment or other environmental insults that are happening. So you can maybe deduce the protein turnover from that snapshot also for the other developmental stages.
Yes, awesome. Professor Jagannathan, I unmuted you. You can speak now. I enabled both your camera and your microphone. Are you able to unmute or?
You can still hear me?
Yeah, I can hear you, Dr.
Okay.
Turck. But I think it's Dr. Jagannathan. I did enable the. It's already enabled, so you should be able to speak, Dr. Jagannathan. Just unmute your mic, and then you should be able to speak. Yeah. Yep. Now we kind of heard something there.
Yeah, the volume is a bit low. Maybe she just has to crank up the volume.
Yeah. Try to unmute the mic, and then you should be able to speak. If not, is there anyone else who has a question? Please type in, or just hit the hand raise button. If not, Dr. Turck, can you put your email ID on the screen if you have it available? If not, it's in the chat.
I can do the chat thing.
Yeah, if you can put it in the chat so that they can directly reach out to you with an email. See if I can. Yeah, I see it here. Okay, I have another question. Oh, yep, perfect. Thank you for placing your email here. I have another question from an attendee. What is the efficiency of labeling and how do you analyze it?
I tried to explain that in the beginning. We have tissue for the 89-day labeling feeding gave us about 95% 15N incorporation. We did this at the time with this QuantiSpec software that relied on MALDI time of flight data. Maybe not everybody has a MALDI mass spec. A good software that I can recommend is the one from John Yates' lab called Census, CENSUS. It's a software that allows you also relative quantification data analysis using the SILAM approach. Yeah, I should check with the Yates lab. The Yates lab was pretty much the one that developed the SILAM, and they have different tools to look for incorporation rates, but also for the relative quantification.
Yep. Okay, sounds good. That is Professor John Yates at Scripps, I believe, right? He's at Scripps right now.
John Yates is at Scripps Research Institute, yes.
Yeah. Okay, we have one last question that I see here. Which mass spec facilities would you recommend for these experiments? How specialized is the analysis?
Yeah, well, again, John Yates comes up. I would think, you know, if you have, if you can entice them to help you, that's where I would go. He has all the software. He has good mass spectrometry. He's one of the top proteomics researchers on the planet. There might be, there's a few others. Michael McCoss at the University of Washington. He used to be in the Yates lab. He has his own lab now at the University of Washington. He's also good. I don't know where you know, Paul Young. I don't know where you're located. In Europe, nobody comes to mind right now. But I mean, theoretically, I think any good mass spectrometry lab can do this. Because the software that you need for the relative quantification, again, is freely available from the Yates lab, the Census software. So you know, you can go to your local facility also. They run your samples, and then you just use the Yates software.
Yeah, Paul is calling in from Ireland.
Ireland, okay.
Somewhere in Europe. Yeah. Okay, sounds good. Yeah, thank you so much, Dr. Turck. We really appreciate your time, and thank you.
Okay.
Thank you everyone for attending. Appreciate it.
Okay, thanks guys. Thanks, Ravi.
Bye bye.
Bye bye.